MS in marine science from Hawaii Pacific University and a PhD in oceanography from Texas A&M University. Her research focuses on climate change and anthropogenic stressors to coral reef ecosystems. And with that, I will turn it over.
Hi, everyone. Happy Aloha Friday. Thanks for having me this afternoon. All right, so the title of my presentation is Building Capacity for Water Quality Research and Monitoring on Maui. Water is one of our most important resources. It's supported along an abundant history of agriculture, both for commercial and private farming. Our oceans house an extensive amount of coral reefs that draw tourists from around the world and help to support our local economy. And then from a cultural perspective, water is really important and historically has helped to shape both kind of social and land divisions. However, our water resources are fragile and limited, and so it's challenging to manage these resources in response to the stressors that come with increased human population and climate change and just human pressures. Huio Kawaiola, which is a citizen science coastal water quality monitoring group on Maui that monitors for coastal water quality on the west and south shores, has reported consistently that turbidity and nutrient levels exceed the standards set by the EPA. The health of our marine ecosystems are declining due to climate change stressors like ocean acidification and ocean warming, and then local stressors like eutrophication and overfishing. And then Maui is geographically isolated and we have limited resources. So monitoring water quality, coastal water quality, or water quality in general on the island is really challenging. Everyone that monitors their water needs to send their samples to Oahu or to the mainland, and that is really time consuming and it's very costly. In response to these challenges, Maui College opened Maui's first water quality lab about a year and a half ago. So we're very new, and as many of you know, it's very time consuming and takes a lot of resources to get a lab up and running, especially to the standards required by some of our partners who have regulatory or compliance testing needs. So we're really now just starting to expand and grow. And we have a few goals focused on education, community, and research. For education, we are providing new educational opportunities for Maui students. So we have two new education grants called Kawaiola and Makuta Makai that are both funded by the USDA. And through these grants, we offer summer bridge courses to high school students in environmental sciences, and trying to kind of get them prepared for their college courses. We've developed new curriculum for Maui College students centered around research and fieldwork experiences in water quality. I'm running a new internship program where we pair our students with government and nonprofit and industry partners to expose them to potential career paths and also get them networking with some of the people on Maui who might give them jobs. And then we also have free intro to water quality courses for just the general community offered through the non-credit side of our college. We also have a NOAA grant called Building Environmental Resiliency Leaders that's funded through the Environmental Literacy Program. And this project has developed new curriculum around community and environmental resilience and is being implemented into high school classrooms around Maui County. We're aiming to build environmental literacy through citizen science. So for one of our research projects, which I'll be talking about in just a little bit, the fieldwork component is really driven by citizen scientists, and And these kind of opportunities help to build community resilience and sustainability, and just gives us a chance as scientists to interact with the community and learn and understand about the marine resource issues that are important to them. We're also increasing capacity for water quality monitoring on Maui. So for example, farms who are regulated under the FSMA, the Food and Safety Modernization Act, need to monitor their ag water for E. coli. And so they collect their samples, they have to store it on ice, and then it needs to be analyzed within 6 hours. So getting that sample over to Oahu is just a real challenge, getting it there in time and analyzed in time. We have National Pollutant Discharge Elimination System permit holders who have to follow strict regulations and guidelines set by the EPA to monitor their water quality. And then we have a number of nonprofit groups who do some science and coastal water quality monitoring on Maui as well. So all of these groups used to have to send their samples off island, and now they can send them to us. So it just makes monitoring for water quality more accessible to our community. And then our last goal is to conduct research that supports healthy coastal ecosystems. So I'm gonna talk briefly about 3 different projects that are all in the preliminary stages, since we're still fairly new, but I'll show you some data and then focus mostly on this last project, reduce eutrophication and acidification in a coral reef ecosystem in West Maui. This first project is an island-wide coastal water quality assessment. This project is funded by the USDA, and the main goal is to establish current baseline coastal water quality on Maui. So we're collecting samples for nutrients, temperature and salinity, turbidity, pH, and oxygen, so kind of all the general water quality parameters. We're doing a spatial assessment, so you can see that we have these transects located on the northwest and into the south side of the island. The transects start as close to shore as possible and then extend out to about 3 kilometers. So we're looking at how water quality varies across the island on different sides of the island, but also what is the extent of land-based sources of pollution? How quickly does that signal get mixed out by different environmental conditions on each side of the island? You'll also notice that there is not any transects on the east side and into the direct south. So there's just, the wind picks up really early there. It's kind of rough. There's just not a lot of access on this side of the island. And so that this whole side of the island will kind of continue to be one of the major gaps in our knowledge for water quality on Maui. We're doing a temporal assessment, so looking at seasonal variability, what is the difference between summer and winter. And then we also received funding to do a large sampling effort in July during the visitor quarantine. So hopefully answering the question of, what is the impact of reduced population and tourism on coastal water quality in Maui. This is a map showing nitrate concentrations, and this data was collected July 2020 when the quarantine was in effect. So, massive reduction in population of about 200,000 people on the island. White is low nitrate concentrations and green is high nitrate concentrations. So right away you can see that the North Shore is associated with higher nitrate concentrations, whereas the West and into the South Shore of the island has low nitrate concentrations. It will be interesting to see if our summer sampling effort, our next summer sampling effort shows the same trends. The west side and the south side of the island is the tourist destination and the resort destination. So, it'll be interesting to see if these kind of trends still hold. This figure is showing silicate concentrations during July 2020 as well, where Light blue is low silicate and dark blue is high silicate. So silicate is a indicator of freshwater, whether it be groundwater or kind of surface waters. So we're seeing higher concentrations of silicate on the North Shore transects. So pairing silicate with another nutrient like nitrate or ammonia can help us understand what the nutrient sources are. So in this case, we have a lot of freshwater sources on the North Shore, so we can link that to nutrient or to nitrate. We do also have some freshwater sources on the west side of the island, despite low nitrate concentrations. So we just finished our winter sampling effort. We'll do another one in the summertime. And just understanding what these trends are across the island and what's driving these trends is really important to our policymakers and our marine resource management efforts on Maui. This second project investigates the role of oceanographic phenomena in the invasive algal outbreak at Manawai. So Manawai is located, it's also known as Pearl and Hermes Atoll, and is located within the northwestern Hawaiian island chain. The northwestern Hawaiian Islands is geographically isolated, it's unpopulated, and one of the largest marine conservation areas in the world. And so a lot of the stressors that we see in the main Hawaiian Islands, like eutrophication and overfishing, we don't see in the northwestern Hawaiian Islands, but there are some stressors that transcend the boundaries of protected areas. And these include climate change stressors like ocean acidification and warming, and also invasive species. So in the case of the chondria outbreak, we don't know how it got there. And I'm not sure we'll ever know, but invasive species can come on the hull of a ship or on someone's dive gear, or through oceanographic processes like currents. This project is funded by NIFWIF and is a collaborative effort between a number of University of Hawaii partners, NOAA, and the College of Charleston. This is a typical Montipora reef at 1 to 2 meters depth at Manawai. You can see visually that it's pretty healthy and happy, and there's not a lot of algal growth. And this is Montipora reef in August of 2019. So now you can see that the reef is covered in these incredibly thick red algal mats. The alga grows alarmingly quickly and just kind of covers everything. They also observed that nothing was eating this red alga, which leads scientists to believe that it's not a native species. So it's just, you can see covering everything. This is what it looks like underneath the mats. So, these mats are really, really thick. The coral's dead underneath, and it smothers out all of the native algae as well. The red alga occurs in high abundance from 1 to at least 21 meters depth. So it was discovered on a routine cruise to the northwestern Hawaiian Islands in August of 2019. I think the ship's crew and the scientists had gone to a number of different locations and then got to Manawai and found the invasive alga, and then couldn't go anywhere else because they didn't want to spread it to any other location. So did their best to survey and collect samples. Allison Sherwood recently and co-authors recently published a paper naming the cryptogenic red alga Chondria tumulosa. It's a new species not observed anywhere else in the Hawaiian archipelago and displays invasive characteristics. John Burns, who is a professor at Hilo, his 3D photogrammetry work captured the alga, and they classified it as an unknown red alga as far back as 2015. So you can't see it, but it's in these yellow circles. And then in 2017 found that cover had increased to greater than 50%. So whether or not it was there before 2015, I'm not sure, but something happened between 2017 and 2019 that allowed this chondria to grow very rapidly. This is a map of Manawai showing the percent cover of the chondria, and this is Heather Spalding's work, who's at the College of Charleston, from surveys in August 2019. So the circles are the shallow reef surveys, and then the squares, which there's not as many of them, are the mesophotic surveys. White is no presence of chondria, and red is about 100% cover. So the chondria is present from the edge of the forereef to the back reef, and all sides of the atoll. It's absent within the lagoon and at mesophotic depths. There doesn't appear to be a strong spatial distribution. It's like a tumbleweed. So if it's in one place, then it kind of spreads rapidly in that location, but might be absent, at least in this survey, on the direct south side of the island. The next steps for this work, we have a cruise going out in July that's dedicated to understanding the systematics, physiological ecology, distribution, and oceanographic processes or conditions that contribute to the spread of the alga. So my lab is working on the oceanographic component. There's not a ton of oceanography and biogeochemistry data or published data at Manawai. One of the things that we're going to be looking at is upwelling. Upwelling is associated with a number of different characteristics, one being high nutrient concentration. So one of the questions that we're asking is, could high nutrient concentrations be contributing to the distribution and presence of this chondria? NOAA's Ecosystem Sciences Division has subsurface temperature recorders, STR, located at various locations around Manawai, and those are shown in the green dots here. And they're located at all different depths. And the recorders have been collecting data for many years now. This figure is showing temperature in degrees C from STRs located on the south, one STR located on the south, which is in black, and an STR located on the north, which is in gray, at 24 meters depth. And it's going back, it's on an annual cycle going back to 2014. So you can see that there's a strong seasonal trend, but there's these very large drops in temperature. And if we just pull out a subsection of the data, here I'm plotting it now on a diel cycle. So you can see now that the temperature drops are anywhere from 6 to 8 degrees C, particularly strong on the south side of the island, and about 2 drops per day, so potentially internal tides. So we're going to be looking at all the historical data to understand the oceanography. We'll also be collecting coral tissue and algal samples for stable isotope analyses, and then just doing a bunch of CBD casts and collecting chlorophyll and carbonate chemistry and nutrient samples as well. Okay, this third project that I'm going to talk about is reduced eutrophication and acidification in a coral reef ecosystem in West Maui. And this is a collaborative effort between my lab and Katie Schamberger and Yina Liu's lab over at Texas A&M University. Kahekili is located on the upper west side of Maui, which is in the Kaanapali area and a popular tourist destination. The Lahaina Wastewater Reclamation Facility receives wastewater for the Lahaina and Kaanapali area, treats that water, and then injects it into injection wells, where it then mixes with groundwater and flows out to the coastal environment, and that water comes out at seeps at Kahekili. The SGD has high nutrient concentrations. It's acidified and potentially has high concentrations of organic pollutants or contaminants. So if you were to go snorkeling at Kahekili, further away from these seeps, you would see that the corals are visually kind of healthy. There's a lot more coral cover. And then if you move progressively towards the SGD seeps, the ecosystem shifts to one that is kind of just more dominated by algae. In response to reef degradation at Kahekili, there have been a number of research efforts. These studies show decreased water quality, algae blooms associated with high nutrient concentrations, and changes in ecosystem composition from coral to algal-dominated reefs. Nancy Prouty's work, which was published in 2017 and 2018, found shifts from net calcification and net photosynthesis to net dissolution and net respiration in corals that were located closer to the seeps as well. Okay, so most of you are probably already familiar with the process of ocean acidification. So atmospheric CO2 dissolves into seawater, reacts with water molecules to form carbonic acid, and then dissociates into bicarbonate and hydrogen ions. So it's the release of these hydrogen ions that lowers pH or increases acidity. Ocean acidification causes a breakdown of the physical reef structure. So this is through the decrease of calcification and an increase in calcium carbonate dissolution. Submarine groundwater discharge also has low pH. And so the influence of SGD can cause what's known as coastal acidification. And coastal acidification has the same effects as ocean acidification. So a decrease in calcification and an increase in calcium carbonate dissolution. In both cases, OA and coastal acidification, the additional hydrogen ions form with these free bicarbonates or carbonates to form more bicarbonate. So ultimately, this process decreases the overall carbonate ion concentration, which is bad for corals because they need calcium and carbonate to form their calcium carbonate structures. So what happens is we get an ecosystem shift from coral to algal-dominated reefs. And this is what's happening at Kahekili. On March 26, 2020, Hawaii's governor issued a 2-week mandatory visitor quarantine that dropped visitor arrivals to Hawaii by almost 98%. And so as I said earlier, a couple 100,000 people, less people on the island of Maui. As a result, the Lahaina wastewater reclamation facility was receiving 40% less injected, or 40% less wastewater, and injecting 60% less treated wastewater. And we were also receiving anecdotal reports of cleaner water around the island. So the question that we've been trying to answer is, can we measure reduced eutrophication and acidification at Kahekili? And if so, how does this impact coral reef health? There's a number of ways to measure coral reef health. So we can do, one of the most common ways is by doing visual surveys, looking at who's on the reef. So how much fish are there? What is the percent coral cover? What is the percent algae cover? We can also look at the health of individual organisms. So putting organisms into tank and doing chemical manipulation experiments, or we can look at the genetics. One of the ways that I study coral reef health is by measuring coral reef metabolism, which is a measure of reef ecosystem health. So how do we measure coral reef metabolism? We collect lots of water samples for carbonate chemistry, specifically total alkalinity, TA, and dissolved inorganic carbon, DIC. And these samples can tell us about organic metabolism, which is the combination of photosynthesis and respiration. And these samples can also tell us about inorganic metabolism, which is calcification and dissolution. And so for this Kahekili study, we're looking at the balance between organic and inorganic metabolism to infer coral reef health. In a healthy reef with low submarine groundwater discharge, the reef's going to be net calcifying, and photosynthesis and respiration will be balanced. Nancy Prouty's work that was published in 2018 found that at periods of high submarine groundwater discharge, which was during low tide, the reef shifted to net dissolution and net respiration. And these are indications of a more stressed or unhealthy coral reef. So we're reproducing this study and comparing our results to this work, the pre-COVID data, to see how metabolism is responding to reduced eutrophication and acidification. We set up some opportunistic field sampling during the quarantine in August 2020, and we just did another large sampling effort in March, 2 months ago. We sampled for 4 consecutive days, 3 times a day, so morning, noon, and evening, and we do the morning, noon, and evening to encompass as much of the metabolic cycle as we can. If we logistically could sample at night, we would do that too. We have 5 different sites. So our vent site is here, and these are all very close to shore, within swimming distance. 3 coral reef sites located at various distances around the vent, and then 1 site down here, which is located further away from the vent. So these sites were all chosen during the Prouty study that was published in 2018. And we're looking at the ratio of inorganic to organic metabolism at each of these sites and comparing that to the pre-COVID ratios collected in the previous study. Just to give you an idea of what these sites look like, this here on the left is the seep site. You can see that there's high algae cover. It's characterized as a dead zone. S2 is a little bit further away, so still very high algae cover, but you're starting to see some corals pop out a little bit. And then S4 is the site that's located the furthest away. And now we're getting less algae cover and higher coral cover. So you can really see how the SGD drives the benthic composition at these sites. And so we can expect metabolism to be different at each of these sites. And so we're comparing the ratio between organic and inorganic metabolism at each site, but also to the pre-COVID data. We've also been doing collecting monthly samples at low tide at the seep here, and then at 2 monitoring wells located on the property of the Kaanapali Ocean Resort. So these monitoring wells access the groundwater. And we're collecting these monthly samples for a couple of reasons. One, to understand the water quality of the groundwater before it mixes with seawater. And then also to track the travel time of the injected wastewater to see, because it travels from the point of injection down to the coast, and to see how it's responding to changes in population. In addition to the carbonate chemistry, TA and DIC, we're also collecting water samples for dissolved inorganic nutrients and PFAS and PPCP, so organic pollutants. And the organic pollutant component is being done by Yina Liu's lab at Texas A&M University. What's really cool about this project is that the fieldwork is driven by citizen scientists. So during our first sampling effort in July, our lab is very new and I didn't really have any students, and we couldn't bring anyone from Texas A&M here because we were in the middle of a quarantine. And so we were fortunate to get connected with the Citizen Science Monitoring Coastal Water Quality Monitoring Group on Maui, Hui Kawai'ola, and they have, and we got them trained, and they've just been really instrumental in helping us to get all of the field work for this project done. So it's just a really great example, I think, of how you can engage the community in your research. Okay, I'll show you some of the data from our nutrients, and we're still waiting on the carbonate chemistry data. So nitrate is on the y-axis and salinity is on the x-axis. And these data were collected at the SEEP site during the Prouty study, which was conducted in 2016. So this is the pre-COVID data. You can see that there's this really nice linear relationship between nitrate and salinity. So low salinity, high nitrate, indication of groundwater, high salinity, and low nitrate concentrations is the seawater end member. And this is our data that was collected at the SEEP site in August 2020, combined with some of our monthly low tide samples. So there's been, we know we're collecting groundwater because we're seeing very low salinity, but the nitrate, there's a major reduction in nitrate of over 90%, 95%. So, we're wondering, is this the effects of reduced nutrient loading due to the population decline? So this was about 5 months post the implementation of the 2-week visitor quarantine. And then these two samples are our most recent SEEP samples collected in the past couple of months. So this is in 2021. Now it looks like the nitrate concentrations might be increasing, but there's only 2 samples. So we'll have to continue to collect our samples to verify this increasing trend. Okay, in summary, the decline in tourism has led to a decline in the amount of wastewater received, treated, and injected by the Lahaina Wastewater Reclamation Facility. And so we're wondering how this affects water quality, specifically nutrients and acidification. So SGD typically leads to eutrophication and acidification, which causes ecosystem shifts from coral to algal-dominated reefs. But with the massive population decline and the decline in tourism, we're wondering if we can measure a response in reef metabolism that indicates a shift to a more healthy ecosystem. And with that, I will be happy to take any questions.
Thank you, Andrea. That was a great presentation. There's a lot that you've done in such a short period of time. I was collecting some questions too. I'll start with Danielle's question. She said, what did you say you were interested in looking at with the chondria species as far as water quality? Hydrogen presence and upwelling?
Yeah, we're looking at upwelling because upwelling is known to happen at Manawai, and that can increase nutrient concentrations. We're collecting samples for total alkalinity and dissolved inorganic carbon, nutrients, chlorophyll, just the regular CTD, cast temperature and salinity, and then coral tissue samples for stable isotope analyses.
Do you bring those back to your lab to analyze them?
No, my lab just does the nutrients,
Okay.
and then we'll be sending out the other samples to UH Manoa and to other locations for analyses.
Awesome. So what I was saying was I actually hit enter on some questions that I was collecting. So one of the questions was, have the citizen scientists caused you to generate any new questions through usage initially when you were doing some of your testing?
Have the citizen scientists caused me to generate any new questions? Um, I, I can't think of any research questions at the top of my head, but just kind of more community, like general questions, like, how do we better involve the community? Like just noticing that it's really important to involve the community in your research, and they can really be advocates of the work that you're doing as well, because they just have such loud voices and they tend to have a lot of time to attend the meetings and things like that with our policymakers. So yeah, just kind of how to better engage the community in research.
Gotcha. I guess that what I was wondering was, as they're doing this work, are they also becoming more engaged in terms of the scientific process, right?
Oh yeah, absolutely. Yeah, for sure, absolutely. They're also like very good at collecting samples. I mean, maybe because they're already really trained, but you find them asking a lot more questions. The tourists are all over the place down in Kaanapali, like for example, for the acidification and nutrient study. So tourists are constantly asking you questions as well, which is just a great opportunity to kind of educate everyone on what the issues are.
And then kind of as a follow-up, what are some of the successes and challenges that you've endured in creating the lab? It seems like a great asset for Maui, but have you come across any really big challenges that you've been able to overcome or still working on? And then, what are the successes that have caused you to really, "Oh, this is great. I'm really glad we have this,"
Yeah, it's until about 3 months ago, it was just me in the lab. And we just hired a lab tech. So that frees up a lot of time to be able to move forward. There's just, I mean, probably a lot of you know, it's just really time consuming to learn new analyses and learn new instrumentation, and then do all of the administrative work that comes with running a lab. So that's been the biggest challenge, and we're growing and getting better every day, and expanding. And then the successes are just being able to work with the community and answer the needs of the community, and their water quality needs.
I also saw that you were measuring PFAS. What was the impetus to begin measuring that? Kind of curious.
Well, I think I don't really know a whole lot about the organic pollutants. That's being done by one of the other professors that we're working with, but the PFAS are indicators of wastewater. And so we're using the PFAS as kind of a tracer for groundwater. So you can use salinity as a tracer for groundwater, you can use nutrients, but those things can kind of change. So PFAS is just another tracer. And then also with the PFAS and the PPCPs, the organic contaminants, we don't really know a whole lot about how these affect coral reef health and metabolism. So it's really just a first-hand look at, can we connect any of these organic pollutants with changes in coral reef metabolism?
Thanks. I think, Dick, do you have a question? You feel free to unmute.
Sorry, I didn't hear your introduction, so this may be totally inappropriate, but I have 3 questions that regard the local community. To what degree are you able to take water that comes up from all the wells on Maui and analyze it for the various herbicides and pesticides that have been used over the years on Maui? And some of that water goes into the drinking supply. Some of it just goes onto the fields, golf courses, etc. That's the first question.
Yeah, we don't do any pesticides or herbicides. We do nutrients, E. coli, and some other just kind of general things. But mainly we're a nutrients lab. A lot of people in the community email me all the time and ask me if I can test their well water for that kind of stuff.
What would it take? What would it take to allow your lab to do that?
A lot of money for instrumentation and someone to learn how to do it, I think. But yeah, I think it's definitely something that people really want to have here. That's just kind of outside of my expertise. And I think to do, to like test, you need to have certain certifications as well, I think, to do those kinds of testing. So
Second question, the Department of Health, State Department of Health has said that upcountry Maui is high priority to convert from cesspools to septic tanks because there's high nitrate levels.
mhm.
Are you able to run tests on the upcountry water system supply to see in fact if their tests were valid? We think that most upcountry residents do not think that they did it properly, and that there are a number of other things that can account for the high levels, the large cattle population and leaching of the manure, all kinds of other issues that are up here. It may have nothing to do with cesspools at all. This is a $200 or $300 million question, because if people country have to convert, we're talking about big money.
Yeah. So you're talking about testing the groundwater or the wastewater?
Well, testing the water that's coming up in the various wells that are up country for
Yeah.
nitrates. That would be
Yeah,
what,
we can test it.
because that's how would the community organize that with your lab to do that?
Yeah, so actually a couple of people had reached out to me about that. I mean, I think that the county, there would have to be some kind of funding effort to do that. But I'm happy to help get involved and facilitate that work, because I know it's really important to the community. I'm also working with the Department of Health and another group in, I think it's the UC Berkeley, the College of Engineering, and they're developing a new alternative system to septic systems that's supposed to be a lot more cost effective, or just like lowering costs for the community. So we're helping them do all of the testing for their pilot study, and it's so far looking pretty promising. So hopefully there will be other options for septic systems.
Last question, quick one. Do you charge if people bring water to you from wells or whatever to say are the nitrates there or whatever? Is there a cost aspect?
Yeah, there's a cost associated with the analyses, and we have a website. If you just Google UHMC Water Quality Lab, all of the costs and the tests that we run are on that website. And then you can just contact me too if you have additional questions.
Thank you.
We had another question from Roy. He had said that you showed in your slides that increased SGD can be increased stress on the coastal ecosystem. He was wondering if you could describe that in a little more detail, and in your own opinion, which marine species end up being most stressed from that, the increased SGD, and I think, what comes along with that, the nutrients.
So the question was, how does increased SGD affect the ecosystem?
Yeah.
Okay. So a number of different ways. I mean, there's been a number of different studies, but specifically I look at coral reef ecosystem health and metabolism. So that's kind of the focus of my research. So when you have increased submarine groundwater discharge, that water has high nutrient concentrations and low pH. It's more acidified. And that causes more respiration of the reef ecosystem, which can increase carbon dioxide levels and then lower the pH, and it drives dissolution. So the combined effect of submarine groundwater discharge is increased respiration and lower rates of calcification, or increased calcium carbonate dissolution, shifts from coral to algal dominated reefs. And then the second question was about the specific organisms, right? That are affected?
Yeah, which were most stressed by the increased SGD?
That's a good question. I mean, I study corals, so I would say corals in general. But I don't really know. As far as like the fish goes, I mean, I know that Kahekili has been established as a herbivory management area. So I know that DLNR looks at the effects of the SGD on the entire ecosystem, including fish and all of that kind of stuff as well, but I'm not really sure how it affects fish. Probably great for algae though.
Maybe herbivorous fish as well.
Yeah. Yeah, there's some overfishing going on over there too. So I know that there was a major decline in the herbivorous fish, but since they've made it into a management area, those fish are coming back and hoping to kind of eat up the algae a little bit more.
This is kind of a follow-up to Dick's question. When you test the drinking water, the private drinking water well supplies, do you have on some of the coastal water quality, we can do delta N15 isotope so that we can kind of isolate which type of nitrogen we're looking at. Do you have any ability to kind of compare, because the ag will have a different weight versus the wastewater?
Yeah, you know what? I don't really know a whole lot about the drinking water and isotopes. It's just kind of outside of my area of expertise. And we would have to send those samples out anyways for the isotope analyses. Yeah, I don't really know.
Well, let me peruse, make sure there was no other questions we missed. I don't have any. Tom, I don't know if you wanted to jump on here and say any last words.
Sure. Thanks everyone for tuning in. This is our last seminar of the semester. Really appreciate you joining us. Want to thank Michael for doing a great job of coordinating this semester's seminar series, and for April and Pat for supporting in the background. And I really want to thank today's speaker. Great job. Really nice to hear about what you're doing, Dr. Kealoha. And thank you for doing so.
Thank you very much.
Well, everyone, thank you.